guys, welcome back. This is part 2 of chapter 2 for the practice test, the practice questions. So up top, concentration tells us the amount of, in a volume of, so remember concentration, we're talking about how much stuff we dissolve in a fluid, right? So how much salt do we have in water for salt water? How much glucose do we have in water for sugar water? The technical terms we use there are solute and solvent. So now you just have to know which is which, right? Which is the salt and which is the water. So concentration tells us the amount of solute in a volume of solvent. Okay? So solute in a volume of solvent. Okay, that's the answer. B has those backwards. C says ions in a volume of solute. No, if that said ions in a volume of solvent, that could be correct, right? That would tell us the concentration, but it doesn't say that, it's backwards. Same thing with D. D says concentration tells us the amount of solvent in a volume of water. So solvent is the water. If this said the amount of solute in a volume of water, that would be correct, right? That would correlate with concentration, but that's backwards, so that's not the answer. And then all of the above, no. Right, so the answer is A. Concentration tells us the amount of solute in a volume of solvent. And there's a lot of ways we could ask this, right? I could give you examples of salt water, sugar water, and I could ask you which is the solute, which is the solvent. Right? So again, always be thinking of ways that the questions could be twisted or asked in a different way, so you can come up with lots of different types of practice questions. Okay, I'm sorry, I keep bumping into this screen and it's a weird movable screen, so it's probably the wavy for you to read. I'm sorry, I'll try and stay away. Which of the following is or are organic molecules? So there you've got to understand what's organic and what's inorganic. Remember, the definition of an organic molecule or an organic compound is something that has both carbon and hydrogen covalently bonded. There's very frequently oxygen, sometimes nitrogen, all different stuff can be mixed in. But in order to be organic, it has to have both carbon and hydrogen covalently bonded together. So let's look at our choices. Sodium chloride, no. We don't see carbon or hydrogen. H2O, or water. No, we have hydrogen, but no carbon. So that is not organic. Both of those are inorganic. Calcium carbonate, so calcium, carbon, and oxygen. It's got carbon, but no hydrogen. So no. C6H12O6. That happens to be the chemical formula for both glucose and fructose, right? Like our basic sugars. Those have carbon and they have hydrogen. So D is the only organic molecule. Okay, so what happens when sodium chloride is put in water? Remember that sodium chloride, or sodium and chlorine are held together with an ionic bond. Right? The sodium is cation, the chlorine is an anion, and those positive and negative charges, the opposite charges attract. So they're held together with an ionic bond. Remember, something special about these ions is that when you put them in water, they dissolve. And I'm sure you've seen this, salt water, right? You put salt in water and it dissolves, the crystals dissolve. That's the sodium and the chloride separated. Or ionizing, disassociating in the water. So let's look at our choices. The sodium and chloride ions dissociate, yes. All right, they disassociate, they separate in the water. The sodium and chloride ions separate and go into solution, yes. Remember, that's what a solution is when you have something dissolves in the water, right? It spreads out evenly throughout the water. That happens with salt in water. The ions are kept in solution because hydration spheres form around them. Yes, Remember, sodium and chloride still have charges. So if you think about it, they could still be attracted to each other. So you've got to wonder, how is it that they separate in water and they don't just link back up together? The reason that they don't, the reason they're kept separate in water is, remember, they get surrounded by water molecules, right? Those are hydration spheres. The water molecules have partial positive and negative areas, right? So they're attracted to the positive charges and negative charges of those ions. If you don't remember that, go back and look at the lecture, because it's kind of a long, involved explanation. But that does happen. So A, B, and C can all occur. That's the answer, right? All of those happen. A hydrophobic molecule. Again, before you look at the choices, think about this. What does hydrophobic mean? Hydro means water, and a phobia is a fear, right? Arachnophobia, fear of spiders. When you have a phobia of something, you have a fear of it. So hydrophobic means water fear. Something that doesn't like water. It doesn't interact with water. It tries to get away from water. We talked about fats and oils, lipids being like this. Again, you mix oil and water, does the oil dissolve nicely? No. The oil all comes together like this to try and get away from the water because it's hydrophobic. So let's look at our choices. A hydrophobic molecule interacts with water? No, doesn't like water. Is polar? No. Likes dissolve likes. Water is polar. So water interacts with or likes polar things, right? Water likes things that are charged, just like it is. So things that don't like water are not charged. They're not polar, right? We call that non-polar, so no. A hydrophobic molecule does not interact with water. Yes, it's afraid of water, right? Hydrophobic, so that's true. Is charged? No, right? We just said water is charged, partially charged. So likes dissolve likes. Water likes things that are charged. Water likes things that are polar. So things that don't like water are not charged. A hydrophobic molecule goes into solution in water. No, it doesn't like the water, okay? So does not interact with water. Now thinking about this, says hydrophobic. I could also say hydrophilic, right? What about a hydrophilic molecule? A hydrophilic molecule does like water. It is polar, or it is charged in some way. It does go into solution in water. Okay, so again, ask the questions multiple ways. Finally, a patient with alkalosis has a pH blank, meaning they have too many or few hydrogen ions. So alkalosis and acidosis have to do with the pH of the blood. The pH of the blood should be between 7.35 and 7.45. Okay? Alkalosis means the blood is too alkaline or too basic. Remember, that's a high pH. So when a pH is higher, it's more alkaline. The blood should be up to 7.45. So if it's above 7.45, it's too alkaline. So let's look at our choices. Patient with alkalosis has a pH greater than 7.45, yes. Greater than 7.45, yes. Less than, no, right? So C and D are not the answer because it's not a pH less than 7.35. So we can cross those up. Let's just look at A and B. So alkalosis is a pH greater than 7.45, meaning they have two. So when the pH is high, does that mean that there are a lot of hydrogen ions or not a lot of hydrogen ions? Remember, hydrogen ion concentration and pH vary in opposite ways. So if the number of hydrogen ion goes down, pH goes up. If the amount of hydrogen ions goes up, pH goes down. So in this case, the blood is too alkaline. The pH is too high. So if the pH went up, that means the concentration of hydrogen ions went down. Okay? So pH greater than 7.45, meaning they have too few hydrogen ions. Okay? The concentration of ions went down. So they have too few hydrogen ions in their blood. That's what made their blood too alkaline. So the answer is A. Speaking of pH, remember that the pH has to do with the concentration of hydrogen ions, right? And that we talk about pH on a scale. We have a pH scale that goes from 0 to 14. Right smack dab in the middle at 7 is neutral, right? That's water. Less than 7 is acidic, greater than 7 is basic or alkaline. So, if a substance has a pH that is greater than 7, it is neutral. No, neutral is right at 7. Acidic? No, we said acidic is less than 7. Basic or alkaline? Yes. A pH greater than 7 is basic or alkaline. So C, let's look at the rest of this. An isotope? Nothing to do with this. A proton donor? No, we didn't go there. Glucose, okay, so now we're talking about carbohydrates, right, like sugars and starches, and glucose is one of the most common, most important sugars in the body. Glucose is stored in the body as, so remember when we have a bunch of glucose, we store it in the body, right, as a much bigger molecule, as a polysaccharide. Just like if you had 1,000,000 pennies, you wouldn't walk around holding 1,000,000 pennies, right? That would be a giant pain. You would convert that into bigger bills. So we do the same thing. If we have a bunch of fuel, so a bunch of glucose, we don't have tons and tons of glucose flowing around the body. We convert that into a much bigger molecule. So we join tons of glucose together and we make a large molecule, and that's how we store glucose. We do the same thing with fats and amino acids, right? We store them as big molecules. Glucose, when we put a bunch of glucose together, that happens to be a molecule called glycogen, right? So we hook a ton of glucose to each other and form glycogen, and we store that mainly in the liver and the muscle. Triglyceride, that's how we store fats, right? Fatty acids. Polypeptide, that's a bunch of amino acids together. So that's how we store amino acids. Fructose is just another sugar, and DNA is made of nucleic acids. Okay, so the answer, we store glucose as glycogen. Carbohydrates, again, carbohydrates are sugars and starches. Carbohydrates are used primarily for what purposes? Again, without even looking at the answers, I know that we use carbohydrates for fuel, right? We use sugars mainly or first to make ATP, our cellular energy. So we use sugars for fuel. To insulate the body, no, that's lipids, right? We have this layer of fat underneath our skin, and that's how we insulate the body. To encode our DNA, no, our DNA is made of nucleotides, right? Nucleic acids go into nucleotides. That's how we make DNA. Carbohydrates are used as the primary structural component of a cell membrane. No, remember that's made out of phospholipids, right? Another type of lipid. Carbohydrates are used as an energy source. Yes, right? We use carbs for energy. As enzymes are proteins, not carbohydrates. So when two monosaccharides undergo a dehydration synthesis reaction, what happens? So first you have to know what a monosaccharide is. Remember, a monosaccharide is one single sugar, right? So one little sugar ring. So glucose is one ring. Fructose is one ring. Mono meaning one. We link sugars together to store them. So when we put two sugars and we link them together, that's now a disaccharide, right? Di meaning two. The way that we link them is via dehydration synthesis, right? Synthesis, we form a bond. The opposite of that, remember, is hydrolysis. So when we want to break them apart again, we use hydrolysis. Okay, so monosaccharides, two simple sugars. When we use dehydration synthesis to link them together, now we have one molecule with two sugar rings. So we form two new monosaccharides. No, we're linking them, right? A disaccharide, yes, right? A disaccharide is two sugars linked together. A polysaccharide is formed. A polysaccharide is when we have many sugars together. So glycogen, for example, that we just talked about, would be an example of a polysaccharide. Okay, so more than two sugars together. A starch is formed? No, starch is a polysaccharide. Starch is a really big polysaccharide. It's not just two sugars. Hydrolysis occurs. Now, hydrolysis is the opposite of dehydration synthesis, so that just doesn't make sense at all. Okay, which of the following is true of lipids? Again, before you look at this, lipids, fats, oils, waxes. Okay, remember these things are hydrophobic. They don't like water. Oil and water don't mix. So these are hydrophobic things. Again, we use them to insulate the body. We can use them for fuel if we don't have sugar available. They include things like cholesterol, steroids, triglycerides, fatty acids, and they are non-polar. So let's look at our answers. Which of the following is true of lipids? Lipids are mainly hydrophobic. Yes, that's true, right? They don't like water. Steroids include a four-ring structure. Yes, that's true. So estrogen, progesterone, cortisol, testosterone, those are all lipids that we call steroid hormones, and they all have one structural feature in common. They're all made up of four rings, right, or kind of like connected circles of carbon. So that's true. Triglycerides include three fatty acid tails linked to a glycerol. That's also true, right? Tri means three. So a triglyceride is a glycerol head and then three tails sticking off of it. A and C, all of the above. Yep, so A, B, and C, all of them were true. Phospholipids have a blank head and a blank tail. Remember, phospholipids are important lipids that make up the plasma membrane. And phospholipids are what we call amphipathic. Amphipathic molecules have two regions that behave differently, right? So phospholipids have this head region that, like water, they interact with water just fine. And then they have these two fatty acid tails that hang down. Those are lipids. Those are hydrophobic. They do not like water. So the two different regions of the molecule behave completely differently from each other. Okay? So phospholipids have a hydrophilic head and a hydrophobic tail. Right? The head likes water, the tail does not. So hydrophilic head, hydrophobic tail, yes, that's true. Acidic and base, this has nothing to do with an acid and base. Right? Same thing with C, nothing to do with an acid and base. Hydrophobic head, no, that's the opposite. Organic head and inorganic tail, no, the whole molecule is an organic molecule. Okay, so the answer is A. Phospholipids have a hydrophilic head and a hydrophobic tail. Now we move on to proteins. Proteins are made from building blocks called, so remember proteins are really, really big organic compounds, and they're made up of smaller building blocks called amino acids. We have 20 different amino acids, and we link them together, right? We string a ton of them together to make a bigger protein, right? Or polypeptide that gets folded into a protein. So proteins are made from building blocks called nucleic acids. That makes DNA, RNA, and ATP. Amino acids, yes. Amino acids make proteins. Fatty acids make triglycerides. Phospholipids, no. Microtubules, no. Almost done. So up top, which protein structure occurs because of hydrogen bonding along the length of the linear chain. So we're talking about protein, and proteins are big and complex. So when we talk about their structure, we talk about their primary structure, secondary structure, tertiary structure, and quaternary structure. Okay, so 1, 2, 3, 4. The primary structure is just this linear chain. So literally just the order of amino acids that we all put in a row. So we can have 80, 100, 200, 300 amino acids in a straight chain. That's the primary structure. Then hydrogen bonding starts to happen. And that straight line will it'll start to bend, right? So it will twist into like a spiral, or it can get folded into pleats, but there's some sort of bending that occurs. That's what this is asking for. So that's the secondary structure. This is the secondary structure. After that, that spiral will get all twisted up into a unique shape. That's tertiary. Then multiple tertiaries all get put together. That's quaternary. There are pictures back in the lecture if you need to see that. Molecules that store and process genetic information are the blank. They're made of blank. So storing and processing genetic information. Right off the bat, you should think of DNA and RNA. DNA contains all of your genetic information. RNA, remember, is how we use that information. How we convert the recipe in the DNA into an actual protein out of the cytoplasm. So DNA and RNA are, so molecules that store and process genetic information are the proteins made out of amino acids? No, that's not genetic information. Are the nucleic acids made out of nucleotides? Yes. So DNA and RNA are nucleic acids. They're made up of smaller building blocks called nucleotides. Nucleic acids made out of amino acids? No, amino acids make proteins. Proteins? No. Proteins? No. So, B. The RNA sequence that's complementary to A, T, C on DNA. So we have DNA, and the nucleotides that we have are A, so adenine, thymine, cytosine. And we need to come up with RNA that matches this. So one, remember that A binds with T, right? T and A, and C and G go together. The other thing you have to remember that in RNA, there is no thymine. There's no T in RNA. So anywhere that you would put a T, you put a U instead, right? You put uracil in RNA. So A would go to T, but there's no T, so that's a U. T goes to A, so UA, C goes to G. So UAG, perfect, UAG. So be able to match those up, right? I'll give you some random sequence, and you should be able to come up with a complementary strand. Last one for chemistry. The structure of RNA differs from DNA. There are a lot of differences between RNA and DNA. Remember, DNA is huge. We keep DNA in the nucleus, and it has the recipes for how we make every protein in the body. Very complex. There's a lot of information in DNA. RNA, we typically just copy a little section, right? Like one little gene. Say I need to make collagen. I'll just copy that section of the DNA. If all I'm making is collagen, I don't need the recipe for all of these other proteins. So RNA is much smaller. Structure, remember DNA is this twisted double helix. So like a ladder that we twist. The sugars are different, right? DNA has deoxyribose, RNA has ribose as the sugar. And then we just talked about thymine and uracil. So DNA has thymine, DNA has thymine, RNA has uracil. So let's look at our choices. RNA contains ribose, yes, right? RNA means ribonucleic acid. Ribo for ribose, so that's true. RNA contains uracil in place of thymine, yes. Everywhere in DNA that we see a thymine, we can't have that thymine in RNA. RNA will have uracil instead. RNA single-stranded? Yes. DNA has 2 strands, right? Double helix, double-stranded. RNA just has 1 strand. DNA contains deoxyribose? Yes. Right? R for ribose, D for deoxyribose. So all of the above are true. All right, that's it. I hope that's helpful. Again, go through, think of other questions, twist these around, see how else you could ask these. And if you're super comfortable with all of this information, you should be pretty good.